Hello everyone. After discuss the basis of the immunology, we got some knowledge about our immune systems. And this lecture, we are going to discuss food and immunology. Nutrition and immunology are at the focus of a scientific revolution. The food supply as a source of nutrients has evolved dramatically over the past 2 centuries. But never before has there been such a promise for innovation. If the food supply is to be improved nutritionally, the needs of the immune system need to be major target of that improvement. The role of the zinc in immunity and inflammation is so critical to be investigated. Zinc is an element that plays important roles in human health. It is not only crucial for maintaining an intact immune system, but is also of vital significance in many other organ systems and processes in the human body. Zinc has a wide range of functions in cellular metabolism, and about 10% of the proteins encoded by human genome have zinc binding motif. Zinc is cofactor for more than 300 enzymes and functions as a catalytic, co-catalytic, structural, or regulatory ion, mainly interacting with amino acids such as histidine, glutamic acid, aspartic acid, and cysteine. It is especially important in highly proliferative systems such as the immune system, as zinc finger motifs can be found in many transcription and replication factors. Consequently, during zinc deficiency, cell proliferation is reduced. The immune system is delicately regulated by zinc. Zinc influences the non-specific innate immunity system as well as the specific adaptive immune system. Zinc deficiency decreased T cell activity could be the major impairment of the effect of IL-2 during zinc deficiency. And what's the role of the vitamins? The role of vitamin A is so critical for immune system. The vitamin A is required in the diet of all vertebrates and is metabolized intracellularly to Ra, which is well known as a regulator of cell proliferation and differentiation. The vitamin A promotes Th2 and restrains Th1 cell differentiation. The vitamin A is an important factor in the differentiation of T regulatory cells, especially in the intestine, with implication for mucous homeostasis and prevention of intestinal inflammatory disorders. The vitamin A regulates lymphocytes humming to the gut, and that dendritic cells, particularly in the intestine, are an important source of Ra for this regulation. Ra may contribute to the antiviral response necessary for defense against measles virus infection. The role of vitamin D and the immune system is so closely connected either. The vitamin D is synthesized in the skin from the action of sunlight. The discovery of VDR, the receptor of vitamin D in cells of the immune system, led to the initial interest in vitamin D as immunoregulatory agent. All immune cells that have been evaluated express the VDR, and in most cell types, activation induces further expression of the VDR. More recently, it has been reported that vitamin D deficiency will associate with an increased incidence of asthma. The effect of vitamin D and the APC is to induce interleukin-10 and suppress IL-12, which has indirect effect on the development of the T-cell subset. The probiotic also function in the immune system. The human gut is a natural habitat for the diverse and dynamic microbial ecosystem, have an important impact on health and disease. Unabsorbable portion of the food are the major nutritional source for this microbial community. Probiotics are non-digestible food ingredients that are selectively fermented in the gut, resulting in specific changes in the composition or activity of gastrointestinal microbiota, thus conferring benefits upon host health. Prebiotics might influence the immune system directly or indirectly, either via specific products derived from carbohydrates fermentation or by altering microbial composition of the gut microbiota. Microbial products such as short-chain fatty acids, defenses, or other bioactive peptides may interact with immune-competent cells, including enterocytes, and modify their status of activation and their activity. Prebiotics have been tested in humans for their ability to prevent infectious disease, as this outcome would be a relevant consequence of improving immune function. Bioactive proteins or food-derived bioactive proteins or peptides show great potential as antimicrobial or immune modulatory and anti-inflammatory effect food. From a nutritional perspective, food protein were once considered only as a supply of amino acids to the body. However, it is now well established that, in addition to their nutrient supply roles, such food proteins in their intact form possess bioactive property, and upon digestion, protein might give rise to a myriad of bioactive peptides. It is becoming increasingly clear that bioactive peptides latently encrypted in the primary sequence of the protein may play an important modulatory role in human body system. Over 20 peptides with antimicrobial activity have been identified following the hydrolysis of casein with digestive proteins. Kappa casein is another source of bioactive peptides with potential in the human health, food safety, and food preservation areas. The peptides derived from beta-casein have been shown to have concentration-dependent effect on mitogen-induced proliferation of human lymphocytes, suppressing proliferation when present at low concentration and enhance it at higher concentrations. The fermented fish protein concentrates led to the enhancement of the peripheral macrophage phagocytosis and increasing IL-4, IL-6, IL-10, or interferon gamma and tumor necrosis factors. Most of these peptides are derived from the hydrolysis of the major milk proteins through proteolysis. The specific effects seen include modulation human lymphocyte proliferation, maturation of T cells and NK cells, enhancement of phagocytic activity, and increased antibody production. The arginine was traditionally classified as a nutritional non-essential amino acid for infant and adult humans. The arginine is a basic amino acid present in physiological fluid. Arginine has many unique metabolic pathways in mammals because of its positively charged group and the side chain. Available evidence shows that physiological levels of arginine are essential to the function of the innate immune system, where mechanisms that involves synthesis of NO and reactive oxygen species, antimicrobial activity, or secretion of hormones, including insulin, growth hormone, prolactin, insulin-like growth factor 1, that regulate the metabolism and activity of immunocytes. And they also could involve in signal transduction pathways via the protein kinase. Physiological level of arginine regulates the adaptive immune system, while the mechanisms that include maturation and proliferation of T lymphocytes and B lymphocytes, the production of cytokine and specific antibodies by lymphocytes and B lymphocytes, respectively, the circulating level of anabolic hormones and the expression of the T cell receptors. And thus, arginine profoundly affects the adaptive immune system in humans and other animals. In support of this notion, clinical studies have shown that enteral and parenteral provision of arginine improves immune function and clinical outcomes in patients with bone injury, with cancer, HIV infection, major traumas, and gastrointestinal surgical operations. Except the proteins, plant foods are also involved in inflammatory process. In summary, based on the number of epidemiological studies, whole grain intake is inversely associated with markers of inflammation. Because brown and jam were also effective, this emphasized the anti-inflammatory potential of the whole grain. Preliminary information suggests a dose-response relationship of whole grain intake and the biomarkers of inflammation. A cross-section study revealed that in healthy elderly subjects, a high intake of vegetable and fruit was associated with significantly lower concentration of CRP. This indicates that high intake of vegetables decreases the risk of chronic inflammation. Vegetable and fruit intake was inversely associated with blood levels of CRP, TNF-alpha, and IL-6. Besides the cross-section studies, several intervention studies have looked at the anti-inflammatory potential of vegetable and fruits under more controlled conditions. The bilberry consumption decreased the plasma concentration of CRP, IL-6, and IL-15, but TNF-alpha was slightly increased compared to the intake of water in subjects with increased risk of cardiovascular disease. LUTs have been part of several dietary intervention studies, which most reported a reduced inflammatory status in subjects consuming such diet. Major contribution to the anti-inflammatory activity of LUTs are polyunsaturated fatty acids and phytochemical, including allylic acid. This is the single mechanism which mediates the anti-inflammatory effects of plant food. Plant food contain many different bioactive components, which all target specific steps or molecules in the process of chronic low-grade inflammation. These bioactive compounds include dietary fiber, PUFA, mineral and trace element, vitamins, and phytochemicals. The knowledge and the practice of the medical use of mushroom originate from traditional Eastern medicine, and in Japanese, China, Korea, and other Asian countries. Clinical practice still utilizes the mushroom-derived preparation. Mushroom-derived dietary beta-glucans have been shown to elicit diverse immunomodulatory effects in human and animal tissues, including the blood, gastrointestinal tract, and spleen. In controlled human trials, beta-glucan intake stimulates the immune system in the blood of healthy adults, adapting the allergic response to the respiratory inflammatory agent, and improved survival in cancer patients. Beta-glucans are chains of D-glucans, polysaccharide, linked by beta-type glycosidic band. The 6-side D-glucose ring can connect to one another on a variety of positions and the D-glucan ring structure. Most beta-glucans are considered to be non-digestible carbohydrates and are fermented to various degrees by the intestinal microbiota. Beta-glucan exerts immunomodulating effect in a variety of species, including humans, other mammals. Their effects are likely mediated via the innate immune system, while binding to a variety of receptors, subsequent release of cytokines, and stimulation to the humoral and cell-mediated parts of the adaptive immune system. Most studies on beta-glucan were carried out in cells and animals. There are limited clinical trials in humans from which we can conclude that beta-glucans are safe and with beneficial effect. Except the benefits from the food to our immune system, there are definitely adverse immune reactions. The term food allergy has been defined as an adverse health effect arising from a specific immune response that occurs reproducibly on exposure to a given food. Food commonly causing food allergy include cow milk, hen's eggs, wheat, peanuts, tree nuts, soy, fish, and shellfish. However, more than 170 different foods have been reported to cause food allergy. Adverse reactions to foods caused by immune mechanisms are often classed as IgE or non-IgE mediated reactions. IgE mediated reactions are those which are immediately type 1 hypersensitivity immune response to food. This slide describes the main immune-mediated adverse reaction to food, including IgE-mediated and non-IgE-mediated allergy. IgE binding and a severe allergic reaction. On such strategy is the synthesis of a short, overlapping allergy peptides sequence which span the entire allergen sequence but are in themselves too short to cause cross-link of specific IgE. Another strategy, again to reduce the risk of IgE binding, have been identified and mutated to the IgE binding site of the allergens, while it's preventing its ability to stimulate T cells. The neutralizing of IgE is another strategy which is still being developed. Anti-human IgE antibody therapy has shown some promise, with the therapy allowing a significant increase in the tolerant dose of peanuts in the patients with peanut allergy. Here is some reference. Thanks for your attention.